Welcome back. In this video, we are going to discuss about the fine structure of eukaryotic chromosome. So under this, we will have an idea about the concept of nucleosome. So let us start the video. An average human cell is around having 6.4 billion base pairs of DNA divided among 46 chromosomes. Okay, 6.4 billion base pairs of DNA which is divided among 46 chromosomes. So obviously the question arises how to fit all the 46 chromosomes into a nucleus which is around 10 micrometer in diameter. Got it? 10 micrometer in diameter. You know that 1 micrometer is equal to 10 to the power minus 6 meter. Got it? So obviously the question arises, so how the 46 chromosomes can be fit into this nucleus which is of such a small diameter, and at the same time maintain DNA in a state which is accessible to enzymes and regulatory proteins? Obviously. So how is it possible that single DNA molecule of each chromosome organized so that it doesn't become tangled with each other? So the answer is that the remarkable manner in which DNA molecule is packaged in the eukaryotic cell. So for solving that, the DNA molecule is packaged into the eukaryotic cell. So from this, the concept of nucleosome arises. Got it? So a single eukaryotic chromosome consists of a single double-stranded DNA. The complexes between eukaryotic DNA and protein is known as chromatin. Got it? And this chromatin having twice as much protein as the DNA. Now, the DNA binding proteins are actually of two categories. One is histone proteins and another one is non-histone proteins. As far as non-histone proteins are concerned, their main job is to help the DNA in replication and gene expression. But histone proteins have got some other role. So what is that? It is obviously positively charged, and it binds to negatively charged DNA molecule. DNA is negatively charged, you know it. So why the histone protein is positively charged? Because it has got high proportion of basic amino acids like arginine and lysine. The histone proteins are of five categories: H1, H2A, H2B, H3 and H4. Got it? These are the five types of histone proteins found in case of DNA. So now you have got an idea about the structure. Now let us know the nucleosome concept. Fact is that the nucleosome concept was first put forward by Roger Kornberg in 1974. He proposed that DNA and histones are organized into repeating subunits called nucleosomes. Okay. According to Roger Kornberg, the DNA and the histones, they are organized into repeating subunits called nucleosomes. And these nucleosomes are considered as the basic structural unit of chromatin. Remember it? Basic structural unit of chromatin. These nucleosomes. So what the nucleosome are made up of? The nucleosome actually consists of a nucleosome core particle and a linker DNA. Okay, we will discuss about what is a nucleosome core particle and what is a linker DNA. Got it? So remember that a nucleosome consists of nucleosome core particle and a linker DNA. So now as far as your nucleosome core particle is concerned, okay, it is actually made up of 146 base pair of DNA, 146 base pair of DNA, which is wrapped 1.67 times around the histone octamer. Okay, so 146 base pair of DNA is wrapped 1.67 times around the histone octamer. So if it is the histone octamer means 2 each from H2A, H2B, H3, H4. So how many? 8 histone proteins. Okay, so there are 8 histone proteins uh, around which 146 base pair of DNA they are wrapped. Okay. The octamer thus consists of consists of four histone proteins. Eight histone proteins, two each from H2A, H2B, H3, and H4. So what about the fifth histone protein? The fifth histone protein, there is presence of a single fifth histone protein, which is bound to the DNA as it enters each nucleosome core particle. So as the DNA enters into nucleosome core particle means here the nucleosome core particle is found. This one is nucleosome core particle where 8 histone proteins are present, 2 each from H2A, H2B, H3 and H4. So 8 histone proteins are present which constitute the histone octamer. So around which there is DNA which is wrapped around 1.67 times. Now as this DNA enters into the nucleosome core particle, it is linked with or it is bound with the H1, the fifth histone protein, which is called H1. Got it? You can see this in the next slide through diagram. So nucleosome, in other words, we can say that, remember, nucleosome and nucleosome core particle, they are different things. So nucleosome is made up of nucleosome core particle along with a linker DNA and H1 bound to DNA as it enters the histone octamers. Got it? So nucleosome core particle is made up of what? 8 histone proteins and a DNA wrapped 1.67 times around the histone octamer. Got it? This is what nucleosome core particle. Now, in addition to nucleosome core particle, there is linker DNA. I will discuss about it. What is linker DNA? And a H1 protein or histone protein which is bound to DNA as it enters the histone octamer. So these three things, they make up the nucleosome. As you can see in this diagram, this is the nucleosome core particle. Okay, this one is the nucleosome core particle. Here there are two molecules each of H2A, H2B, H3, H4. So how many histone proteins are present? 8 histone proteins are present. Okay. Now these 8 histone proteins, they constitute the histone octamer. Now around these 8 histone proteins, the DNA is wrapped. This is DNA. The DNA is wrapped how many times? 1.67 times. 1.67 times. Got it? This is what your nucleosome core particle. This is nucleosome core particle. So now this nucleosome core particle is also attached with H1. How? The H1 actually gets bound to the DNA as this one is DNA as it enters into the nucleosome core particle. As it wraps around the nucleosome core particle, at the beginning the H1 is bound to this DNA. Okay, so you can see in this diagram, this one is the nucleosome core particle, this one is the nucleosome core particle, this one is the DNA which wraps around the nucleosome core particle. Then this one is the histone H1 which is bound to the DNA. This is the DNA. As the DNA is entering or is wrapped around the nucleosome core particle, this H1 is bound to it. Got it? In addition that, so you can say that this one is the nucleosome core particle, this one is the nucleosome core particle. Two nucleosome core particles are attached by means of a linker DNA. This is the linker DNA. Okay? Linker DNA. Now this is what your nucleosome consists of. So nucleosome consists of a nucleosome core particle, the H1 protein and the linker DNA. In this linker DNA, the non-histone proteins are attached. Got it? So this is very simple as far as nucleosome concept is concerned. And this nucleosome is considered as the basic unit of chromatin. Okay? Now, let us know about the different labels of packaging. You know that the DNA needs to be packed well so that it can fit into the smaller diameter, smaller sized nucleus, which is around 10 micrometer. in diameter. So there are we are going to study about three levels of packaging in case of DNA. What are those? First one is a 10 nanometer fiber. Then the second one, that means the 10 nanometer fiber further condenses into a 30 nanometer solenoid. Okay. Then the last one is a the 30 nanometer fiber, 30 nanometer solenoid further condenses into a metaphasic chromosome, which reduces the the original size of the DNA by 10,000 fold. Okay, so three levels of packaging we are going to study. What are those? Remember that the first new formation of nucleosome is the first level of packaging of DNA into a chromosome. Got it? It results in the formation of a 10 nanometer fiber, which under electron microscope appears beaded. Now, the packaging of the DNA into a 10 nanometer chromatin fiber, which we are calling as nucleosome, shortens the length by around 6-fold. Okay. Now, this chromatin fiber further condenses, that is the 10 nanometer fiber further condenses into a 30 nanometer fiber. That results in the total condensation of about 50-fold. This 30 nanometer fiber is called solenoid. Now, in the interphase, most of the chromatin, which we otherwise known as euchromatin, is in solenoid form, that is in a 30 nanometer fiber condition. Okay. Now, as the cell is entering into the mitosis, the chromosomes become highly condensed so that they can be distributed to daughter cells. Now, loops of 30 nanometer chromatin fibers fold upon themselves to form a metaphase chromosome, where DNA is condensed nearly 10,000 fold. Okay. So metaphasic chromosome is organized into large loops attached to protein scaffold. Got it? So three levels of packaging. First one is a 10 nanometer fiber, which is reducing the size by around 6-fold. Then further condensation to form a 30 nanometer fiber, which is reducing the size into by 50-fold. Now this 13 nanometer fiber, that is called solenoid, further condenses into the metaphasic chromosome, which is reducing the size by around 10,000 fold. Okay, in this way the large size DNA is packed into a nucleus which is of very small size. Okay, I hope this video would be helpful for you in understanding the fine structure of eukaryotic chromosome and the nucleosome concept. Thank you.